I'm here for the presentation for the Introduction to Biochemistry, and I'm myself Divya Divakaran, Assistant Professor, Department of Pharmaceutical Chemistry from Yenepoya Pharmacy College and Research Center. So here we'll be discussing on the key biochemical concepts and application of techniques, analyzing metabolic pathways, evaluating enzyme functions and their kinetics, interpreting molecular biology principles, integrating biochemistry in the healthcare and disease management, and communicate biochemical pathways. So coming to the introduction to biochemistry, biochemistry is a study of the chemical process that occur within the living organisms. It explores the fundamental molecules and reactions that sustains our daily life from the tiniest microbes to multicellular organisms. So coming to the historical background of biochemistry, in the 18th century, Antony Lavoisier's work on fermentation was the notable one. Coming to the year 1828, Friedrich Wöhler synthesized urea from ammonium cyanide. Coming to the Louis Pasteur's works, he demonstrated the role in fermentations, and Wilhelm Kuhne coined the term enzyme in the year 1878, and Edward Buchner worked on the cell-free fermentation, who won Nobel Prize for that. And coming to the year 1937, Hans Krebs elucidated the citric acid cycle, which is central to energy production, earning a Nobel Prize in the same. In the year 1953, discovery of DNA's double helix structure by Watson and Crick marked a revolutionary moment in the molecular biology. The field advanced further recombinant DNA technology in the year 1970, laying the foundation for the modern research in genomics and proteomics, shaping biochemistry as a cornerstone of science. The main applications are in field of medicines, disease diagnosis, where biomarkers are used for conditions like diabetes, cancer and genetic disorders. In case of drug development, targeted therapies where biochemical pathways are utilized. Molecular genetics, study of genetic mutations causing hereditary disease are validated. Clinical biochemistry, monitoring patient health through blood, urine or tissue analysis. Coming to the industrial application, biotechnology mainly utilized in biofuels, enzymes and bioplastic productions. Coming to the food industry, fermentation, nutritional analysis and preservation techniques. In pharmaceutical industry, mainly in the production of antibiotics, vaccines and hormones like insulin, and in cosmetics, mainly in antioxidant products. Other areas like nutrition, forensic sciences. Coming to the major biomolecules in the biochemistry as proteins, lipids, carbohydrate, nucleic acids, etc. Proteins are the workhouse of cell, responsible for catalyzing reactions, providing structural support and facilitating the growth. Lipids includes fats, oils and plays a crucial role in the energy storage in membranes and also in signaling processes. Carbohydrates are the primary source of energy for cells and also for all other tissues and for signaling molecules. Nucleic acids comprising DNA and RNA are the biomolecules which are essential for the genetic information storage and also their expression, and made of nucleotides like sugar, phosphate and nitrogenous bases. DNA forms a helix while RNA is a single-stranded. DNA stores hereditary data and it plays a very crucial role in the advancement in the biotechnology. Coming to some of the enzymatic reactions and metabolic pathways, enzymes are biological catalysts that accelerate the chemical reactions, allowing cells to maintain the high reaction rate which is very necessary for the lives to sustain. Regulation of them, biochemical processes are tightly regulated to maintain the homeostasis and respond to changing environmental and physiological conditions. Metabolic pathways are interconnection between the enzyme catalytes and the breakdown of these to synthesize major biomolecules important for the life. Certain techniques tools which are utilized in biochemistry are chromatographic technique, spectroscopic technique, electrophoresis and other techniques. In chromatography, it includes gas chromatography, high performance liquid chromatography, thin layer chromatography, which mainly separates complex mixtures into individual components and mainly applied in analyzing biomolecules, identifying compounds, proteins and nucleic acid purification. Coming to the spectroscopic techniques, mainly includes UV, infrared nuclear magnetic resonance which is NMR, and mass spectroscopy which is mainly used in studies used in the studies of interactions between light and the matter, and mainly utilized in the determining molecular structure, concentration, functional groups, and mainly NMR is utilized for the organic compounds. Electrophoresis is mainly utilized in separating molecules based on their size and charge under an electric field, which mainly comprises of gel electrophoresis and capillary electrophoresis, and used in DNA fingerprinting, protein analysis, and diagnostic testing. Other techniques includes Western blotting, polymerase chain reactions, and mainly utilized in the DNA amplification for genetic studies and diagnostics, and mainly this techniques includes with specialized application in biochemistry. And certain cellular respirations like glycolysis, citric acid cycle, and electron transport chain, which mainly includes in the metabolism of the biomolecules. Glycolysis, which is initial stage of cellular respiration, where glucose is broken down into pyruvate or lactate, which produces ATP, and later this will be gone into the citric acid cycle, which is also known as the Krebs cycles, which is the major cyclic pathway, further oxidizes the pyruvate into to get more ATP. Electron transport chain is the final stage where this ATP will be passed to a series of complex to generate a large amount of ATP required for the life to sustain. Photosynthesis, that is light reactions and Calvin cycle. There are light reactions which is light dependent reactions where it uses sunlight, energy of sunlight, and uses to generate ATP and NADH. And there is Calvin cycle, which is also known as dark reactions, where it is light independent cycle and uses the ATP and NADH to convert carbon dioxide into organic compounds. And we have Calvin cycle are used in the synthesis of glucose, the primary energy source for the cells. And there are certain factors which regulate the biochemical process like allosteric regulation, transcriptional control, feedback loops, and post-translational modifications. Allosteric enzymes can be regulated by binding a small molecule to the other, so there is a site other than the active site where the allosteric molecules will be coming and binding, and which then regulates or controls the normal enzymatic process by which we can regulate the enzyme actions, and gene expressions can be regulated to control the production of enzymes and other proteins. So certain applications of biochemistry in the medicine and technology mainly includes in genetic engineering, diagnostic test, drug development, and biofuels. In genetic engineering, the manipulating DNA and proteins to create new organisms or therapies, and in case of diagnostic test, it analyzes biochemical markers to detect or monitor diseases, and in drug development, it is mainly designs new drug by understanding biochemical mechanisms, and in case of biofuels, using enzymes, microorganisms to produce new fuels. And recap, we have covered the fundamental concepts and applications of biochemistry from biomolecules, the smaller particle, to the major metabolic pathways, and this plays an important role in the future where it mainly applied in the biochemical industries, pharmaceutical industries, and major research goals. Thank you.